Okay, that should be good. Usually the recording goes pretty flawlessly, but you never know. So we'll see how it goes. Okay, so we'll start with this. Reading the entire genome code. It should say genetic code, but anyways. Listen to this part. A, G, G. Isn't this exciting? He was like, yay, isn't this so exciting? I don't want you to read the textbook like this, where it's like you're just reading all of this stuff for no apparent reason. I want you to find the exciting parts of it and read those things, and hopefully the quizzes will help you there. Okay, so I'm going to start with transcription, and I'm not going to teach you. Okay, don't even take notes. Stop. Why are you taking notes? Who told you to come to class and take notes? Someone probably told you. I just want to remind you, it's all recorded. It's all online. You can go back. How many of you can think and write at the same time? Okay, well, I can't. I have no ability to do this and use these at the same time. It's not possible. So if you can think and write at the same time, feel free. But I would encourage you to jot down topics, keywords, stuff like that, things that you can go back to when you listen to the recordings. Do not feel the need to write down everything I say because it's going to be there online for you. So relax, listen, enjoy. Now, if you want to write, you can. I won't stop you. Okay, so transcription. So I'm not going to teach you the mechanism of transcription. I'll be doing that in 409 in the spring. But I want to start with just introducing transcription as just a starting point for cell biology. And why am I saying transcription? Well, first of all, what is transcription? What's the purpose of transcription? You had it on the pre-test. Raise your hand. I'll start calling random people next week. Yes?
It converts DNA into RNA.
It converts DNA into RNA. Does it convert all the DNA into RNA? No. What? So which DNA is converted into RNA? An intron?
Oh, the whatever genes are required to respond to an external signal.
Right, so whatever genes are required to respond to whatever signal. Right. Right. So, and so in terms of introns and exons, the thing to always remember is the exons are expressed, the introns are spliced out. But we won't even go there right now. Right, and it's regulated, right? Because it's not like the cell's not going to just be like, hey, we'll need gene A today. There's some reason that certain genes are expressed, right? In response to some kind of signal. Right, so I want you to start there, because I want you to take a minute to just think about that the fact that the cell needs to survive in its environment. It needs to communicate with other cells in the area, either as a cell floating through the blood or as part of an organ or as part of a culture if we're talking bacteria, right? So that it needs to respond to its environment. And if you try to understand cell biology in the context of that, then you should be good, okay? Okay, so transcription is regulated, so it happens in response to some stimuli. How is it regulated? What does that mean? Those of you who took 409, I see you guys. You know this answer. How is it? What does that mean? What is Dr. Ehrlich talking about? Regulated. Come on, throw some words out. I know, God, it's like, so it's the afternoon, we don't want to talk. Yes? Some kind of feedback, so it's happening in response to something. Let's get more specific. What does regulate to mean? When you regulate your thermostat, what are you doing?
Keeping it under control.
You're controlling it, you're turning it on and off, you're keeping it somewhere, you're changing it. So same with gene expression, when you regulate something, you're turning it on or off, you're keeping it somewhere, right? And your thermostat is in response to your temperature in your house and in your body, right? But in a cell, it's in response to the environment. Okay, so we're regulating, okay. Proteins bind to, remember there are proteins involved. Gene expression is regulated by proteins that come and bind to promoters or cancer agents and activate gene expression. Does anyone know what that means? Are you guys like, it's still summertime? Why are you talking about proteins? Okay. Okay, so you have this external stimulus. It says, hey, I'm a cytokine, I'm binding to a receptor, and all this signaling happens, and a protein is activated in some way, usually through phosphorylation or something like that, and it goes into the nucleus, it binds to part of the DNA, and helps to recruit RNA polymerase, which then what does RNA polymerase do? Nothing? It what? It binds to DNA and does what? You guys just don't want to jump out there. It's too late. I hear whispering over here. Does RNA polymerase do something? Does it copy the something into the something? Yes.
It moves down the DNA template strand. It starts to.
Okay, so it goes down the DNA template strand and starts to do what? Make something? RNA? Makes RNA? Okay, you guys. Come on, I gotta start teaching this at 9 in the morning again. Okay. Then at least I can tell you to go get a coffee. But I feel like coffee at this hour, you've been up all night. Okay, anyways, let's move on. Okay. No, this is its nucleus, not its cell phone. Okay, so in a eukaryotic cell at least, there's a nucleus, and a lot of this is happening in the nucleus, but obviously for a eukaryotic cell, it's not, there's no nucleus. It's actually just happening in the area around the DNA. Okay? Okay, so where is this all happening, right? Just like what I said, in eukaryotic cells, you have a nucleus, and in prokaryotic cells, you don't, right? Okay, so you've got transcription, remember we're doing transcription, and we're talking about gene expression. I'm bringing you up to speed. So now you've got RNA made. And so in eukaryotic cells, which I'm partial to, mRNA is exported from the nucleus, and only mature mRNA is exported from the nucleus. What's mature mRNA? What does that mean? Does that mean you're 18 or 21? Yes?
It has the cap and the polyethylene.
Right, so it's been capped, it's been polyadenylated, and what else has it been? Spliced. Introns are removed. Great. Okay, so it says it right here, so if you didn't know, you can read it. So, all right, so why is mRNA capped, spliced, and polyadenylated? Yes? Okay, stability is one reason, you're right, because if you wanted to destabilize an RNA, decapping and removing its polyA tail are a great way to destabilize an RNA. Yep? Doesn't it increase the abundance? So that's kind of related to what he says. It increases the abundance of a particular protein because it helps to stabilize the RNA. And also, what else does it do?
Prevents degradation.
Again, prevents degradation, stabilize RNA. Yep.
It makes translation easier.
It makes translation easier. Okay, good, because it helps for bringing, well, it helps for association with ribosome. What else? There's one big one. Yep.
So that the only way you can get out of the nucleus.
Right, right, exactly. So only capped and polyadenylated and spliced RNAs can get out of the nucleus. Right. So those are all right things. Right. So if you're trying to isolate mRNA from the cell, what would you look for? So you're a biotech company. You are each your own biotech company. And you need to develop a technology to isolate mRNA only from eukaryotic cells. What would you look for? What would you develop? Do you want to talk? Let's talk to our friends for a minute. So you all know the person you're sitting next to. So talk to each other for a minute and come up with a method to isolate mRNA from eukaryotic and prokaryotic cells. You got about 2 minutes. Come up with your business plan. Okay, so does everyone know their neighbor? It seems like you guys know each other, so that's good. I see some lonely people up there, so make sure you take time to get to know your neighbors. Okay, so if you were trying, let's start with the easy one. If you're trying to isolate mRNA from eukaryotic cells, how would you do it? Yes?
You look for the cap and the poly-A tail.
Right, so you develop some way, something that will bind to the cap and poly-A tail, wash, everything else goes away. Yeah, that's good, that's an easy one. Great, any other ideas? Okay, well let's go with that, right? So a little bit harder, prokaryotic cell, what would you do? I actually have to think about this one a little myself. Yeah.
Like you look for ones with uracil.
So that's right, uracil. That was the only thing I can come up with also. That's the difference between, well, and that's really going to be a hard one because, so it could be uracil and something else, right? Because when you're trying to isolate mRNA, you're trying to differentiate between mRNA and DNA, but also and other RNAs like ribosomal RNA and other small RNAs. So it would have to probably be size also. And that's, so actually there's some company, I was trying to think about how to do this, and there's a company where they have a kit where they'll, where you can isolate bacterial RNA from infected eukaryotic cells. And so I was like, oh, of course it's all proprietary, but from what I can get, basically what they try to do is get rid of all the other stuff, and what you're left with is the bacterial RNA. So you can bind up all your poly-A and all your caps and all that stuff and get rid of that, and then you're left with your prokaryotic. Okay, that was fun, wasn't it? Okay, so our mature mRNA is exported from the nucleus because there are these, I don't like these legs. Here we go, that's better. Because there are these poly-A binding proteins and cap binding proteins, cap complex, and there's an RNA export factor, and all these things bind it and help to get it out of the nucleus, and then there are some things that remain in the nucleus because they need to export future RNAs. And then the RNA is going to associate with translation initiation factors, and translation begins. And, well, we already know how the cell knows which RNA to export because we just talked about that. Okay, so we've got RNA, it's out of the nucleus, if you're a eukaryotic cell. And then what do you need? You need translation to happen if you're an mRNA, right? Do all RNAs get translated? No, right, okay. What kind of RNA does not get translated? tRNA. What else? All right, ribosomal RNA, what else? All the other ones? SI, MI, small nuclear, small nucleolar RNAs, all of the RNAs involved in the RNAs involved in splicing. Okay, there's a lot of RNAs. We all have to acknowledge the fact there's a lot more than just mRNA. Good. So then you have translation. Okay, so there are many ribosomes that are present in the cytosol, and they're either free or associated with the ER. We spoke about that last week, why that is, right? Why are they associated with the ER? Because these are proteins that are going to be entering the secretory pathway. And so this right here is actually showing a cross-section of these ribosomes sitting on the ER membrane. So that's your rough ER. And so ribosomes are made of protein and RNA. It's actually mostly made of RNA. Proteins are really just there to hold their RNA together in the right conformation. And then you get translation of your mRNA, and that's based on the genetic code, right? You know, you have your triplet code, your codons that encode which amino acids will be added. What's the effect of an insertion or deletion in the DNA or RNA? Yep. A frame shift, right. Right, because you know you have to have the triplet code, and if you pop one in, it's going to completely shift the frame. Exactly. So each codon has a tRNA, another RNA. And you have your anticodon that's going to recognize, it's going to base pair with your codon, and then you've got the amino acid attached up here. Okay, so translation, it starts, and you have translation, and then it stops. As you can tell, this is more of an overview, right? So what codon starts translation? AG or ATG? Oh. Hello. Right, so all proteins start with methionine, right? And then there's stop codons that signal the end, and there's more than one stop codon. So how does the ribosome know which AUG to start at? Does anyone know? Because you can guess, you've got how many bases? How many bases are there in RNA? 1, 4, good. So just by chance alone, you're likely to come upon many AUGs, right? Just by chance. So how does the ribosome know which AUG to start at? Let's see, yeah. Let's try something. Ta-ta box is something else. What else? Kozak sequence in eukaryotic cells. And what's it called in prokaryotic cells? Shine-Delgarno. It's okay. Basically, it's the context in which the AUG is in. It's the bases immediately surrounding the AUG. I'm not going to make you memorize it because you can look it up. But it's a very important thing to know because when you go on out into the world and become scientists, somewhere along the way, unless you're an ecologist, and you probably won't have to do this, but if you're a cell or molecular biologist, somewhere along the way is going to say, hey, I really want to clone this gene and express it as a protein. Can you do that? And you'll say, what? But you'll figure it out. And one thing I want you all to remember, because I screwed this up before, is include your Kozak sequence. Because if you don't put your Kozak sequence in, what might happen? It won't start at the right place. If you've got a whole string of bases, right, in your RNA, and you don't start at the right AUG, are you going to get anything resembling the right protein? No, because your frame is going to be totally different. Yes?
Does each protein have a unique Kozak sequence, or is it the same?
Kozak sequence is a general string of bases. So it's pretty much the same thing. There's like one or two that could be an either or, but it's like something, 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 AUG something. But those few somethings are pretty standard. And that would make sense, right? Because the same ribosome is going to find it. Yeah, good question. Yeah, so the question was, does each protein have its own cosec sequence? And no, it's pretty much universal because the ribosome needs to recognize it. Good question. Okay, so you've identified your correct start because you remembered to put the cosec sequence in, or nature has remembered to put a cosec sequence there. And then you have your ribosome assembles, and your first tRNA comes in with your methionine, and then you add each additional tRNA on according to the codon that's present, and you get the growth of your protein, your polypeptide chain. And then when you hit your stop codon, what amino acid is added? None, because you're stopping. And actually a water molecule is added. Yeah, it's true.
It's true.
Okay, so who knew that many ribosomes bind to one RNA? Who knew that? Okay, someone did. That's good. A few people did. And who knew that many RNA polymerases jump onto the same gene and start transcription at the same time? So it's great. Good. Some people know. That's great. So this was one of those things that was kind of mind-blowing to me when I started to understand it. But you know, you've got your transcription, you've got your gene there, and you've got your transcriptional start site, and then you've got your stop, right? And then you'll have your RNA polymerase bind and start transcription. Well, as soon as that RNA polymerase clears that start site, another one's going to come on, and another one's going to come on, and you're going to end up with tons and tons and tons of RNA from one gene, right? Because you'll just make one copy. That's not very useful, is it? Let's say you need to upregulate a gene like crazy, like IL-2 is involved in cell growth in response to an infection. You want a lot of IL-2 so that all those immune cells can grow and fight an infection. So you want a lot of RNA and a lot of protein. So making one copy of RNA and one protein is not very useful, right? So I want you to think of things in a larger scale. You're going to have lots of copies of RNA when a gene is expressed very highly. And then these ribosomes are going to assemble. They're going to find the start site. They're going to move this way and then start making the protein. And then another ribosome is going to hop on the same RNA right behind it. And so that's a polyribosome. So you'll have one mRNA, many ribosomes, lots of protein. Okay? I'm kind of just flying through here. Does anyone want to add anything? Anyone have any questions, thoughts, feelings? No. Okay. Yeah. So we've got gene expression, we've got transcription, we've got translation. Now what? Okay. We've transcribed, we've translated, we've got protein, lots and lots of protein. What has to happen to the polypeptide chain? We just hang out and we go home, put class is over? Yep. It's got to fold. Right. Right. What else does it have to do? Where does it need to go? Wherever it's supposed to go. I guess there's okay. Exactly. Every protein is like, proteins are like people. Sorry, I crack myself up all the time. They're like people. They have their own jobs. They do their own thing. Right? They go to work. I work in the nucleus. Right? They need to go where they need to go to do their job. Because every protein has a job. Right? And what do? I don't know. Whatever proteins do. Right? Because that's protein specific. I can't really answer that one. So what if there's a mistake? What if there's a mistake in the folding? Then what? What can happen?
It's destroyed.
Gets destroyed. What happens if misfolded proteins don't get destroyed?
Can they cause other proteins to misfold?
Huh?
Can they cause other proteins to misfold?
They can cause other proteins to misfold.
Prions in the brain.
Oh, prions. Oh. Those are just bad proteins. Well, let's say, okay, prions make all sorts of trouble. Absolutely. Let's talk about plain old proteins. What if hemoglobin doesn't fold properly? What will it cause? Sickle cell anemia. That's one thing. That's one thing. Yeah, it will cause anemia because it probably won't bind iron very well. Right. So what's the point of all of this? Well, so this was my small introduction to the course. So the first thing we'll talk about is intracellular traffic. You've got this protein and it needs to go somewhere, it needs to do something, right? Misfolded proteins are associated with disease, not just horrible prions, but for example, misfolded proteins form Lewy bodies in Parkinson's disease. They're also associated with Alzheimer's, diabetes, cystic fibrosis, some cancers. We want our proteins to fold correctly, and when they don't, we want them to be degraded. Dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. That's one of my favorites. So that's my introduction to cell cycle. We'll talk a lot about the cell cycle, how there's a fixed amount of time for completion of each event. Each event, what's each event? As in M phase, G1, S, G2, M phase, right? Each event needs to proceed in the correct order. They each need to occur once per cycle, right? 2 mitoses in a row, and that's what. Each event must have a start and an end, and there must be backup mechanisms to make sure that cell cycle proceeds normally in case there's some sort of issue. And then cell cycle needs to be adaptable, right? Because is the cell cycle the same in skin cells and in neurons? No, it's going to be on its own cycle because there are different cell types that need to divide based on their environment and what they're doing. Right. And then there are checkpoints, and this was in the pretest, right? So checkpoints are making sure everything's going properly, and if a checkpoint is activated because something is not happening correctly, then it halts the cell cycle. So that things can hopefully be fixed. And if they can't be fixed, then what happens, or what should happen? Apoptosis, right. So for example, there's a checkpoint that monitors attachment of chromosomes to the mitotic spindle, right? So what happens if your chromosomes are not properly attached to, or not attached to your mitotic spindle? What happens to the cell? What happens to the daughter cells? Who's saying? Someone raise your hand. I hear Right, uneven number of chromosomes. Right, right. And is that good for the organism? No. What happens when we don't divide properly and we have the wrong chromosomes going to the wrong places? That's a hallmark of cancer. That's one of those things that you see. Another thing is if your DNA is not completely replicated because of DNA damage, that also is going to activate a checkpoint and inhibit beginning of mitosis. So these are all points to ensure that the cell cycle does not progress if conditions are not favorable or if there's damage. Right, so what regulates the cell cycle? I just heard someone say CDKs. Yeah? CDKs and what? cyclins, right? So there are proteins that regulate the cell cycle. And the cells and the amounts of proteins go up and down, and that is what determines if you're entering a phase of the cell cycle or you're going to end a phase of the cell cycle, generally. What happens if the cell cycle is not regulated? Cancer. That's my favorite. When in doubt, say cancer. Right. Someone once, so people get to know me really quickly, and they realize I'm always telling you you're going to get cancer, and I'm always talking about ubiquitin, which that hasn't started much yet here, but it will. And I think someone on an exam once was like, I'm not sure, but ubiquitin, cancer. They didn't get any points. So if you have a dysregulated cell cycle, we have apoptosis. And it's interesting because often a really good cancer cell, like a cancer cell that's really good at being a cancer cell, what are two properties of a really successful cancer cell? Yes? Uncontrolled growth. And what's the other one? Okay, there's one. Well, high mutation rate, but related to apoptosis?
It looks like normal cells.
Looks like normal cells. What did I just hear? Evasion. Okay, so evasion of P53 doesn't work. We'll get very deeply into that later. So what I'm looking for, who wants to add to this? I'm getting more, I want a more general, yes. They don't die. Exactly. They don't die. They're really good at growing and really bad at dying. That's a perfect cancer cell. Right. So this is going to be Unit 4. I'm not going in order. This is going to be Unit 4, where we're going to talk about controlling cell numbers. Here's where you screwed up. Telophase occurs after metaphase. Right? You don't want to. You got to keep that in order, right? Okay. Okay. So this is just another pretty picture because I love immunofluorescence. Again, your DAPI is always blue, and that's always your nucleus because DAPI means DNA. So this is just showing M phase. So your DNA is going to condense, and then you're going to get metaphase. Remember, anaphase, late anaphase, telophase, right? So metaphase, telophase. Okay. So now we're going to segue into the cytoskeleton because we do a nice unit on cytoskeleton. And cytoskeleton, people always, whenever we talk about cytoskeleton, in the beginning, people always tell me, well, it's so this cell is strong and it can move. But it's a lot more than that. So what role does the cytoskeleton play in mitosis? Mitotic spindle, right? That's made of microtubules, right? So how could an understanding of the role of the cytoskeleton in mitosis lead to cancer drug development? Do you guys want to talk about that for a minute with your friends? Or you on it? I know where you see hands going up. Do you guys want to take a minute? Yeah. Let's take a minute because sometimes that breaks up a nice long class. Talk to each other. How could the understanding of the role of the cytoskeleton in mitosis lead to cancer drug development? Okay. Everyone, it's like this massive quieting down. So y'all have made your first millions in cancer drug development. So how could an understanding of the role of cytoskeleton in mitosis lead to cancer drug development? Who wants to? Give me one. I saw you before. Yep. That's the most obvious one, right? So if you inhibit the mitotic spindle, then you can't get cell division, right? And actually, I believe that's what Taxol does, right? And we'll talk about that when we start to study the cytoskeleton. Anyone want to add to that? Anyone have a novel idea? Because Taxol's out there already, but if we can develop a new drug, I think we can be friends. New cancer drug. Anyone? Anyone? Yeah, you get an A. If you get me a patent for a new cancer drug, an A. Okay.
Keep your A, I'll keep my A.
What'd you say?
I said you can keep your A, I'll keep mine.
There you go. Okay. So that's Unit 3, cell structure and function, where we talk about the cytoskeleton and how it regulates the function of the cell. Yes?
So there's a drug already on the market for something like this.
Yeah.
How do they isolate just the cancer cells and not the rest of the body cells?
So the question is, okay, so there's a drug already on the market to inhibit the mitotic spindle microtubule, but yeah, how are you not killing the rest of the cells in the body? So the way most cancer drugs work is they target the cells that divide the fastest. And so since your cancer cells are dividing the fastest, then they're going to be more sensitive to these drugs. But yes, there's crazy toxicity, which is why people on chemo are not happy people usually. Yeah. Good question. Okay. Okay, so now we're really going to start. So that was just fun. Have fun. Okay, someone smiled. Good, thank you. You have to give that to me every now and then. Okay, so Unit 1 is intracellular. I hope this is what we're doing. Yep. Unit 1 is intracellular traffic. Sometimes I just don't even know what I'm doing. Okay, so we're talking DNA to functional proteins. So pretty much that's what we just went over. We started with transcription, but that's essentially going from DNA to RNA, and then we went from RNA to protein, and then functional protein. Right, so we get transcription, translation, and then proteins need to fold, right? This you all know, primary, secondary, tertiary, quaternary. Proteins need to get to the right place, right? So if you work in the ER, you need to get to the ER. If you work in the nucleus, you need to get to the nucleus. Gene expression is regulated by external signals. So let's talk a little bit more about this. So this is what we mean. These are all different. This is right out of the textbook. These are all different cell types. So you've got prostates. This is a microarray. Who knows what a microarray is? Sometimes you have to stop me when I start putting stuff up that looks like nothing to you. What's a microarray? Yep, in the corner. So you can look, so you have something with, so you can look genome-wide, right? And you're looking at gene expression, global gene expression. And you compare, here what they're doing is comparing different cell types. So if it's red, it's up. If it's green, it's down. If it's black, it's no change. And so what you're seeing here is the whole thing at once. But if we zoomed in, you would see a teeny little square in each spot all the way down. So here's prostate cancer, and all the way down is the whole genome. And here's lung cancer, and all the way down is the whole genome. Brain, leukemia, renal, stomach, ovarian, breast, liver. And what you can see, so in each spot along here, it's the same gene. Same gene. And what you can see is there's some crazy difference between different cells and what genes are being expressed. So for example, here you see some green in these cells, and look, here it's all red. So here you're getting lower expression of whatever gene this is in these cancers, and in these cancers, you're getting higher expression. And so this is just to kind of drive home that concept that gene expression is cell type specific. So what kind of genes do you think are going to be expressed differentially in different cell types? Just what's your favorite? Okay, let's start easier than that. What kind of genes are going to be expressed the same in all cell types? Maybe, okay, how about this? You don't have to give me a gene name, but how about a process that it might be involved with? Yep. Huh? I can't hear you. Shout it out. Cell division related genes, maybe, because all cells need to divide, but actually since they divide differently at different time scales, it might kind of change a little bit. But you're going in the right direction of a process that all cells have to do. So how about metabolic processes? Might also change depending on how frequent you're dividing. But these kind of like normal homeostasis genes tend to be the same across, like actin. Actin. All cells are pretty much expressing actin. Or tubulin. Yes?
I have a question about that. Is that information like multiple, like groups of people, or just like...
Yeah, so here so you can imagine if you were to zoom in on this, it would look like this. Where each so this would be patient 1, patient 2, patient 3, patient 4, and then that would be gene 1, gene 2, gene 3, gene 4, like that. Right. So it's like, which is why you see these clusters of color, but what you can see is that they are cancer specific. So this is all prostate cancer. So this region right here, these tend to be upregulated all the time in prostate cancer and liver cancer.
So is that all like, is that like a full, somebody's full genome, like just one chromosome?
Whole gene, this is a whole genome. Whole genome. Microarrays are very powerful, but they're very scary because they give you so much data and you have to figure out how to handle it, which is why I push you all to go into bioinformatics. Every single one of you. But because that's what you need. You need to know how to program to handle this kind of data, or you need to know someone who knows how to program, or use the right software. So the main point, though, is so for example, so genes involved in secreting estrogen. So estrogen is going to be expressed differently in a ovary versus a, I don't know, skin, right? Just to make it very, very basic, right? So there's differences in different cell types, and that's because they're doing different things. Okay, and so different cells are expressing different proteins. So this is also straight out of the book. You can go look at it. And this is looking at proteins coming out of brain and liver. And this is a 2D gel. A two-dimensional gel. Who's seen a 2D gel before? So someone raise a hand. Anyone want to tell me? Oh, so who knows what SDS page is? Yeah. What's the purpose of SDS page? Say it in the most basic way possible. So to separate proteins, right? And separate proteins based on what? Size, right. Right. And so with a 2D gel, you're separating in the one dimension. First is isoelectric point, so that's this way. And then the second way is this by size. So you've got your acidic and your basic proteins, and then up top here are large proteins, at the bottom are small proteins. So isoelectric point and molecular weight. And just the purpose is to get good separation. The purpose of this figure here is just to show you that the cells express different proteins. And so what I want you to point out is, so the red actually is supposed to be proteins that are in common between the two samples, and blue is supposed to be proteins that are different. But if you'll notice, the abundance is very different, right? So you might have some of the same proteins. So for example, see this little dot here? And that little bigger dot there, or you've got this dot. There's 2 dots here, and there's 1 dot there. So you're going to get differences in the amount of protein made, depending on your cell type, because different cells are going to need different amounts of protein. And then you'll also get, notice you'll get these horizontal dots. And so those are usually the same protein that's been modified in some way. What kind of modifications happen on proteins? Post-translational modifications. Huh?
Adding a sugar.
Like, so you can have sugar, so like glycosylation. What else can happen? Yep. Methylation, acetylation. Acetylation happens on proteins. Is methylation, I guess. Methylation, I think of DNA, but yep. Phosphorylation, all the Asians. Right? What other Asians do you have? Phosphorylation, ubiquination, simulation, metylation, isolation, all the Asians. So these are all ways that proteins are modified that help to affect their function, or that totally affect their function. And so you can see here, see how there's 1, 2, 3, 4, 5 spots in a row? And the only difference is they're all the same molecular weight, but the only difference is their isoelectric point, and they're getting more acidic as you go to the left. So it's likely that this has been modified in some way that's affected charge. So different cells express different proteins that are modified differently. And this is all because cells do different things and need proteins that are regulated to do different things at different times in different places. Okay? Okay. We just talked about this. Okay, so we talked about genes that would be differentially expressed in different cell types, and genes that you'd express that would be uniformly expressed in normal cells. What kind of genes do you think would be expressed differently in cancer cells? What might be expressed really, really high in cancer cells? Yes. Cell cycle?
Yeah.
Genes that would promote cell cycle. So perhaps cyclins, right? Or CDKs. What other genes? What genes might be expressed really low in cancer cells? Who was raising their hand? You were raising your hand? No. Were you raising your hand?
P53 gets knocked out in most cancer cells.
Right, P53, so that's the second time I heard P53. P53 often is not functional in cancer cells. So P53 should be expressed very low in cells that are functioning normally. But it's kind of a red flag for when things are not functioning normally, and it starts to build up and activates all these things to shut off cell division and activate apoptosis and all these things to stop cells from dividing uncontrolled. And in many cancers, that gets knocked out, so it's essentially low, and it kind of tricks the cell into thinking things are normal. Great. Anyone want to add to that? Okay. What about an infected cell? If you're an infected cell, infected with a virus or bacteria, why not? What kind of genes might be expressed really high? And you don't need to give me a gene name, you can give me a process. Come on, common sense. Yep.
Involved in replication and transcription.
Oh, genes involved in transcription or DNA replication because the virus uses it? Okay. Okay. I like that. Great. And what else? What other genes might be expressed really high in an infected cell? Huh?
Immune genes.
Immune genes, exactly. So you guys are getting the point here, right? Okay. So here's something to think about. So 10,000 to 20,000 of the cell's 30,000 genes are expressed at any given moment. But the ultimate picture of protein expression is much larger. Take into account post-transcriptional regulation and post-translational modifications effect on protein function. So let's think about that just for a minute. So you might have 1 to 2/3 of all genes being expressed, but there's many more proteins that come from that. Okay? You guys need to wrap your brains around that, or you're like, well, duh. Okay, what's post-transcriptional regulation? What does that mean? What's an example of some kind of post-transcriptional process that might result in multiple proteins? I've seen the same few people. 1, 2, 3. Come on, give me someone new. Give me someone new. I haven't seen you that much. Yeah.
We did splicing.
Splicing. Okay. Who wants to add to that? Yep.
RNA editing.
RNA editing. Okay. So we've got alternative splicing. We've got RNA editing. What else? Anything else? We'll go through a few things. Those are 2 good ones. And then there are post-translational modifications, and these are all these simulation, phosphorylation. Those are covalent modifications on a protein that are going to affect its function. So this results in an expansion of the functional proteome. That's what you can do. You've got your genome, but your genome does not reflect your functional proteome in any way, right? Because your functional proteome means your proteins that are able to do something, that actually function in doing something. You might have 1 gene, which might result in 10 proteins, which will be modified in 20 different ways, which will do 50 million different things. So, functional proteome. Okay, so this is what we're talking about. So you can have, you can get regulation at many different points. You can get regulation in terms of transcriptional control, so whether or not a gene is transcribed or not, right? You can get regulation in terms of RNA processing control, so alternative splicing, stuff like that, RNA editing. You can get regulation in terms of RNA transport and localization. So some RNAs are transported to certain sites in the cell where they're going to function. And then you'll get translation, and you can get whether or not the protein is translated at all. Sometimes you'll get inhibition of translation. Sometimes you'll get degradation of the RNA if you don't want any protein to be made, as if you have a choice. It's really yourself controlling this. So then you'll have mRNA degradation. So if you've got no mRNA, you're not going to get any protein. If you don't have translation happening, then you're not going to get any protein. So that's another way to kind of shut things off. And then if you do get protein, then you can get all these modifications that will affect whether or not a protein is going to go to a certain place or interact with another protein or be stable or be degraded. And then when you want to shut something off, what do? You want to shut off a process? What's the best way to shut something off? You want to shut off M phase, you degrade. So remember, so when you degrade proteins that are involved in regulating certain processes, you're shutting off that process. So when it's time to turn off a process, you can ubiquitinate and target that protein that's critical to doing whatever job that needs to be done in the cell. You can target that protein to be degraded, and then it shuts off the process. Okay? Okay, so we just went through this. That's it for now. So I'm going to let you guys go early, those who want to go home and celebrate Rosh Hashanah, enjoy. And I will see you all Monday. Monday, don't forget, there's recitation beginning. Okay? If you guys needed to talk to me quickly about getting on Blackboard to take the free test on now.